Hello, my name is Hannah Lichtenstein. I've been working in Dr. Catherine Lenz's lab for the past 3 years, and here's my poster on early life stress effects on limbic microglia phagocytosis in male and female rats. So let's start off with some background. Adverse childhood experiences, or ACEs for short, have shown an exposure response relationship to mood disorders in adulthood. So this means that children who go through more ACEs are more likely to be prone to the development of mood disorders such as anxiety and depression. Previous rodent models of ACEs, such as childhood abuse, have been created via maternal separation stress, wherein pups are separated from their mother for a specific period each day. So this maternal separation stress model activates the hypothalamic pituitary adrenal axis, or the HPA axis, which is the stress response. So after repeated activation of this system during development, stressed animals presented anxiety-like behaviors and impaired social interaction during adolescence and adulthood. This early life stress may activate microglia, and those are the cells that we're interested in this study. And these are the immune cells of the brain, and they're crucial for developmental processes like myelination, axon guidance, synaptic pruning, hippocampal phagocytosis of neural progenitor cells, and neurogenesis. There's a lot of things that microglia are responsible for. But in response to stress, microglia activate the immune response, and they signal phagocytosis through increased inflammatory signaling. This inflammatory response can be sustained throughout adulthood in animals that experience early life stress. Developmental stress may disrupt or unbalance these developmental processes, and this can lead to suboptimal circuit organization and maturation, and that can place the organism at risk for cognitive and behavioral dysfunction, such as mood and anxiety disorders. And in previous studies, interestingly, male rats have been subjected to chronic restraint stress, and they exhibited increased density of microglia in the medial prefrontal cortex and the nucleus accumbens, which are two areas that are relevant to affective and mood disorders. And in this study, we were looking at limbic regions of the brain. So the goal was to determine if early life stress had effects on limbic microglia phagocytosis, and if this effect differs in male and female Sprague-Dawley rats. So the limbic area of the brain was selected for this study, which includes areas like the hippocampus, amygdala, and hypothalamus, because this area of the brain is really heavily impacted in not only the stress response, but also in mood disorders and social behavior. All right, so here's some information about the animals and the methods that we use. So we ordered 8 pregnant female Sprague-Dawley rats, and the day of birth for each pup litter was designated as postnatal day 0 or PD0. So pups were then stressed via maternal separation stress for 4 hours per day from day PD2 to 20 during the light cycle, as this is the time of day that most maternal care occurs. So this maternal behavior was observed for 10 minutes before handling, and again once the pups were returned. So half of the pups were randomly selected at PD21 for tissue collection, and then they were post-fixed and brain hemispheres and were sectioned at 45 micrometer thickness. So these pups, half of the brains were processed for qPCR analysis of gene expression, and we were looking at microglia phagocytosis in all our regions of interest. And then the other half of those brains were processed for immunofluorescence staining, or IF staining, and this lets us visualize microglia as well as CD68 positive microglia in stereo investigator. We also did statistical analysis in GraphPad Prism 7, and we used a two-way ANOVA with a p-value of 0.05. The Bonferroni post-hoc test was conducted after the two-way ANOVA, and this adjusted the p-value for multiple comparisons. We also used R and RStudio to convert qPCR fold changes into aggregate z-scores. First off, stress increased hippocampal expression of genes related to microglia phagocytosis in both sexes, but there was no effect on these genes in the hypothalamus or amygdala. We only found significant effects in the hippocampus. And our specific genes of interest were ThyroBP, C1qa, CD68, and CyBB. ThyroBP showed the most significant effects in the hippocampus, and these effects were not shown in these other genes of interest. And in previous studies, ThyroBP has been shown to enhance microglia phagocytosis. Here are images of the dentate gyrus, CA1 and CA3 regions of the hippocampus that we imaged via immunofluorescence staining. Based on our preliminary results from gene expression in the limbic regions, IF staining was first conducted in the hippocampus. The dentate gyrus, CA1 and CA3 regions of the hippocampus were all imaged after staining. And after analyzing the hypothalamus and amygdala via IF staining, we found that there were no statistically significant effects. So in these images, the microglia are shown in red, CD68 positive microglia, which signifies phagocytosis, are double stained with green, and then the blue signifies cell bodies. So the first row shows animals in the control condition, and the second row shows animals in the stress condition. Then we used these images to conduct some experimental analysis, and we found that stress led to a significant increase in the percent area stained by microglia in the dentate gyrus, a trending increase in the CA1 and no effect in the CA3. And then when we examined the effects of stress or sex on the number of microglia in just any region of the hippocampus, we didn't find any significant effects. Moving forward, so we wanted to look at CD68 positive microglia. CD68 binds to the lysosomes of macrophages, and therefore it's a really good marker of phagocytic activity, wherein microglia that are CD68 positive are engaging in phagocytic activity. So males seem to exhibit a higher percentage of CD68 positive microglia independent of stress in the DG, and there were no effects in the CA1 or CA3. And then post hoc analysis indicated that the main effect was actually driven by a baseline sex difference in the control animals. So this is really interesting because previous studies have actually found that at earlier ages, females have more microglia phagocytosis than males, and males have higher microglia phagocytosis later in development. So this suggests that male microglia may be more vulnerable to activation at this time in development, and that lines up with the previous literature. So here are some conclusions and future directions for this project. Based on the results, early life stress seems to impact microglia phagocytic activity in the hippocampus more than the hypothalamus or the amygdala. And this is really interesting because the hippocampus is a crucial regulator of mood, social behavior, and the stress response. And all of these things are dysregulated in mood disorders like anxiety and depression. Therefore, stress-related microglia phagocytic changes in the hippocampus via early life stress may be contributing to stress-related programming of adult behavior and cognitive functions. So future studies should maybe consider other time points such as adolescence, as this is a critical period in development, and it can be interesting to see the comparison in these animals among different time points in development. And future studies should also investigate other types of early life stress, as these results are not necessarily generalizable to all forms of early life stress, since we only use maternal separation stress as a model. And then future studies should also determine what are the potential targets of this phagocytic activity. So one idea that I've had is that a potential target could be perineuronal nets. So that is what I'm working on for my thesis, which I will be defending in the spring. And here are my works cited. So if you have any questions, please feel free to send me a message or contact me regarding this, and I'd be happy to answer any questions that you may have. Thank you for coming to my presentation. Have a great day.